Hello. Hi. Good to see everyone. I'm Monica, just in case you haven't met me yet, Monica Poelchau. I think I've met most all of you so far. So, and do you want to, so I don't know who all needs to be at this meeting. So how about you all just check to make sure that everyone's here who needs to be here, and then you can give me the go ahead.
I'm not sure, but I think let's go because there's much stuff to discuss.
Sure. Okay, great. Well, and I see you're recording this meeting, so that's great. So yeah, I'm Monica, and thanks for joining me for this session. I'll start sharing my screen. It says host disabled participant screen sharing.
Apologies, let me fix this. It should be good now.
All right, let me go. Okay, great. All right, so hopefully you can see my screen with a PowerPoint, and there's a Firefox tab behind it. So are we good there?
Yep.
Yep, okay, great. So today we were going to talk specifically about some BPRI specific information that I thought would be helpful for you to have with your project specific goals and deliverables. And before I started setting up this training, I reread Fringy's very helpful email summary from our previous discussion, where we went over some of the annotation Jamboree goals and outcomes that you all had defined. And thanks for putting that together, Fringy, so I don't have to. So just to remind ourselves, there's an educational goal that we're trying to meet here, making sure that everyone is educated how to look at a genome with a question. You're going to make a tree, generate a phylogenetic insight, and perhaps do some other things there. And there's an integration goal to integrate the genomes in a phylogenomic manner. And as outcomes, we hope to have different groups with different biological interests. They'll each have a list of 5 to 20 genes. And again, this phylogenetic tree educational goal, okay? So I was keeping this in mind as I was trying to think about what you all would need to help to do this process most efficiently. And I'm not sure if this was articulated, I think it was, but this also said to me that you might have a mix of publication outcomes where some of it might be, some undergraduates who are content to have the experience of creating a phylogenetic tree and writing up a report, and it will end there. But other outcomes might be that you're going to write publications, peer-reviewed scientific publications about this, where there's going to be a bit higher bar in terms of data management and quality of the annotation results. So I wanted to go with you all to address those things and a couple other background points here. So that formed the agenda, which I'm going to talk about just basics. So how should people sign up? What data sets are available at the I5K workspace, in Apollo, JBrowse, at NCBI, which is where RefSeq generated the annotation data. I'm going to show you I would recommend, or some pathways to finding information about schistosarca genomes. At NCBI and I5K, I took the liberty of finding 2 genes in that spreadsheet that you all have of Orco and Ebony, just to see what's around, just to see what hurdles you might encounter. I'm not going to go into comprehensive detail, because if you are going to train at looking, finding a gene family, looking at phylogenetic trees, you're going to probably find other ways of finding the data, but I want to go over these as well. Also, because the path here for this group is a bit different than for our other users, since we don't have that, BLAST link between our BLAST and Apollo. And there's chat. Oh, great. I didn't know that page. I should probably check that out. Thanks, Ocean. Indeed. And then, so a couple other things. So naming is one. So when we think about, where we're going to go with the annotations at the end, and if we want to publish them and submit them to NCBI for better data reuse, Then naming is the big thing that takes a lot of my time. And so I thought it'd be worthwhile investing some time in it to explaining this to you. So so that it is, we have less of a log jams in that area. So things can move forward more quickly. And I want to talk a bit about those annotation outcomes that were defined by sort of your goals for this project too. And then, Q&A. So I'm hoping I'm going to cover many of the gaps that you might have that are not defined in the other webinars that I sent, the recordings that just go over how Apollo works and how to, make a basic annotation, evaluate RNA-seq data, that kind of thing. But please, during this webinar or afterwards, whenever, just raise your hand and use the chat, whatever, so we can sort of figure out where things are missing. And if need be, we can also set up another session. So just to refer to some of the other training resources that exist for the Apollo basics, and I sent you those in the email that I had previously, I think. Okay, so very much basic. Please ask your new annotators to register at our registration link. That's super basic. I can show you where to find that. If you don't want to go to that URL, you go to annotate here on our homepage, click that and just add some super basic information and select the organisms that you want to register for. It will go faster if usually I have to approve, or our policy is that I ask the genome owner before I approve people. So if you have a list of people that that aren't in this meeting here that need accounts, just go ahead and send me an email list so we can just get them through faster. Okay, data sets. So I wanted to show you just, I should have known about your link that you have there, Hoju, and I'll check that out later. Maybe I should check that out now. But I wanted to also just walk you through some of our resources at the I5K workspace so you can just see where things are. So I'm going to show you Schistosarca americana. I'm going to share these slides with you later so you have the direct links to everything. But basically, just to find these things, you can go to organisms and find your favorite locust or grasshoppers. So here's Schistosarca americana. This is our basic organism page that just shows you a description of what the grasshopper or locust is. And here in this analysis section is where the most interesting information for you probably is about the data sets that are available. So the RefSeq information, functional annotations. So we use a pipeline to generate gene ontology and KEGG annotations for the proteins for each data set. And I'll show you, I just want to point out the link to that information there too, in case this is useful for working with your annotators. Sometimes some people like to have that information if you're interested in a pathway or a particular gene ontology category. You can use what we've already computed there. Oh, thanks. And of course, the genome assembly. So just to find that, you can click on this link for the functional annotation page. And we have the source URI here is where the data is. So you can go find that data set here. I'm not going to go into detail about how this was generated. I can do that later. And we have another webinar and slides for that as well. Okay, so RNA-seq. So Maeva provided us with RNA-seq information. And Amanda aligned that to the genome. So thanks to both of you. So to find these things, you can go to Tools, JBrowse Apollo, or use the direct link that I gave you here. Find the genome browser, and go Schistosarca americana. That will load. And so right now we have the NCBI gene models. There we go. The NCBI gene models, GC content gaps, and the coverage plots plus junctions. And the combination of those two should give you decent information about the where things are expressed. So here, for example, and if you want to know where it came from or what SRA accessions or SRX accessions were used, you can click down here and go to About This Track, and it should show you where those came from outside of the spreadsheet that Maeva shared. I will point out, we still don't have anything for gregaria in terms of RNA-seq, so we need to get that information from you before your full annotation effort starts.
I thought we were going to do the ISO-seq at the minimum. I know that the other data hasn't come in, the Illumina data, but I thought we were gonna align the ISOSEQ data.
Yes, we are going to do that. I'm just going to send everything at the same time so that it's done in one shot for Gregaria, if it's okay.
Oh, okay. I didn't know if it may be just as easy, Monica, for you guys to run the processes as they come. Or I guess it's Amanda, what do you feel?
Yeah, I guess Amanda, it's up to you. Do you want to do in piecemeal?
So yeah, I guess they'll end up being separate tracks. So it'll be 2 runs either way. I assume.
Yeah.
So
The
yeah, it
ISOSEQ
probably
gets
doesn't make
7 different tissues, so I guess we have to decide if you're gonna pool it into one track or keep it
Yeah.
separate. But yeah.
I suspect it doesn't make a huge difference. Do whatever is convenient for you. Just make sure you tell me exactly how you want it displayed and which samples go where. And that should work for me.
Well, I just figure it gets some data in sooner and other data may follow because I know it takes you guys a little while to upload things.
Yeah.
Great. Thank you both.
Yeah.
Okay, great. And then there was also talk of generating ortholog alignments, so protein alignments from other species. So if you want to have those displayed as well, we'd also need those, a few weeks before you start in earnest. And I forget exactly when it is that you're starting in earnest, apologies, but soonish, right? So, but it's not, it would be helpful for the annotation effort. I don't consider it strictly necessary if that's just not possible at this time anymore. So, but do let us know if you do want to load those things as well. And then finally, I'm sure you all know this based on what Hojun sent, but there's also just the information that is available at NCBI for the different Schistocerca species, where if you have really detailed questions about where those gene models came from, what data was used to generate the gene predictions, you can find that at NCBI. So, okay, basics. So now I wanted to go first into finding things, and I'm going to spend a little bit of time about finding things based on text because I want to explore that too. So say, for example, I want to find Orco, odorant receptor, co-receptor in all my species. Can I just search for Orco at NCBI? I thought, well, maybe that's a decent way, because you can do that for fly data, actually. It's well indexed and well described enough that that's possible. So the short of it is, like, I don't recommend it because you're not going to be able to find things comprehensively that way at NCBI. You can try, but basically what I found is that the source databases that it comes from, so GenBank, which is just a user submitted thing, so it really depends on what the user decided to call that data is what you can search, and then whatever RefSeq decided to call it. And so one thing that you might want to do that could be an outcome of this exercise here is to actually improve the metadata that we send to NCBI, because then that can be indexed and retrieved by future annotators or future people using this data. So you can go to the protein search, but I did not recommend going this route. Instead, just do sequence-based for the basic single copy ortholog search. You all know how to do BLAST, so I don't want to insult your intelligence with this. I'm not going to show you the NCBI BLAST just because I'm sure you all know it, but I just tried out and found the fly Drosophila melanogaster record for odorant receptor, co-receptor, and NCBI. It's pretty easy to find. You get that protein sequence and the results. You can do that now, but that gives you a more comprehensive set of results from those databases. I will show you using our I5K BLAST how that works. So if we go here's our BLAST page. You can also find this from our homepage just by going to the tools menu and going to BLAST there. But if we just look here at finding Schistocerca americana, and hang on, I think the protein sequence. Yep. I'm just going to cheat because I have the protein sequence already up and here. And so this, the databases that you saw that were available, oh, great. Gonna have to. So if you find this, we have a bug that I'll need to talk to our team about for our blast that gives you spurious false results. So email me if you find that, but that's not what's supposed to happen. So, and if it happens, you can also just give the same query a couple of tries. So I apologize about that. So essentially the databases that we have available are all just the RefSeq data, the proteins, coding sequence, and transcripts. So here I'm going to blast against the protein sequences there. If you went to NCBI to do this, you'd also have the externally submitted Schista-Circa data from GenBank as well. This is the curse of the live demo. Apologize about this. Okay, there we go. Yeah, I'm sorry. So this is not working and I'll have to text Chris to reset stuff. We don't really need this for our setup right here. So it's not going to impede the functionality of the webinar. But basically what you would see if you saw the results is that you'd have a list of results and you'd get what you need from that information is the accession numbers. And I'll show you how to work with those in a bit here. So here I have a slide for that. You get that accession number from NCBI, and that's going to be, XP, an XP number, and you can search for that in the search box here. So, and I will show you that as well. So here, for example, I can just start typing in an XP identifier, which ideally you'd paste in and have from our blast search. And that will pop you right into that area that you need in JBrowser Apollo. Okay. All right, so next up, the next try I did this with your data is Ebony. So
hang on a minute, sorry, Monica. Going back to the browser, that was the Orca gene?
No, this is not. So if you want to find that, so that I just typed in a random.
Oh, okay.
Sorry,
Right.
yeah, this is not. But yeah, here we have that XP. I have that in the slides here. Sorry, I do not have that canned right now. So
No worries.
But the other thing that you can do is if you know what it's called by RefSeq. So in this case, since I did a bit of background work, I know that it's not called Orco. I know that it's called odor receptor co-receptor. And so there's a bunch of them right here. Odor receptor, this one has 2 isoforms. There's a couple that RefSeq appended with the -like, which means that it found sufficient similarity to the odorant receptor co-receptor, but not enough. So here you can also search that way, but it's not a fuzzy search. So you have to have the exact string, the exact name to find it. And if you put in, sometimes you do odorant receptor co-receptor, that will get you to the wrong area too. So that's why the sequence-based search is really best. And so here, this gets us to the correct area in JBrowse. I see there's things in the chat. Okay.
Hey, Monica, I just did it. I'm on a Americana Apollo, and I just typed in hexamerin, and it says hexamerin-like, and then it pops up another window that says hexamerin-like is found in multiple locations. What is that about?
So here, let me just show this for everyone.
Yeah.
So yeah, it's in multiple locations. So it would be, they all seem to be on this this sequence, this scaffold, but they have different coordinates. So looks like there's multiple copies, possibly paralogs, on the same scaffold. So we go to show, it will pop us there. If you hit go, I think it closes this window and just sets you over there. If we click show here, it will pop you to the next one. So you can sort of course through all the different coordinates here.
I see.
Any other questions? Nope. Okay. Great. I'm getting a spinning wheel. Apologies. Okay, here we go. So again, if we try to do this sort of search again, this time with ebony, this time I can show you I'm at the NCBI protein search page. So you can just, see what's available and type in ebony. And the cliff notes is this is going to show you nothing. So. Okay. So, and again, we could try using the same BLAST approach with the fly ebony sequence. So I'd suggest fly is the reference organism that I chose, but I'm not a Schistocerca biologist, so I don't know if there's better resources available to, do that homology-based search from a better annotated reference organism. So that would be up to you.
So we can ask you that. I mean, this is fairly basal when it comes to flying insects. So the nearest things would be a cockroach or a termite. Is that right, Ho-Jun? And
Yes.
I don't think we've got a good annotation of either of those. I don't know about termites. And then if I go completely basal to flying insects, probably the next big thing that comes to mind is Daphnia. And I'm kind of thinking that maybe it's best to just stick to Drosophila. Do you know of anything else that's well annotated? Like, is it worth putting in a track for Daphnia proteins?
I haven't kept up with the quality of the Daphnia assembly. I mean, the ones that I'm familiar with, were they from first generation, from even before then? So
Yeah, me too.
The quality was just not what we would expect and want nowadays. And so therefore the annotation, the assembly quality, and therefore the annotation quality, we can expect to be subpar as well. So yeah, fly might still be better. But then again, I haven't kept up to date on Daphnia to see if they have any of these, if they have assemblies with some of these newer technologies like the locusts that provide complete assemblies, and therefore we can assume that at least the structures themselves are in much better shape.
Yeah. So my sense is, sticking with well annotated insect genome, whether it's Drosophila or a honeybee or tribolium, and those things will work fine. Unfortunately, we don't really have anything that's well annotated in Paonioptera. When we go to sort of non-holometallous insects, I don't know, aphid, some hemitron might have a decent annotation, but they will compare with Drosophila anyways. So,
Is there a good honeybee? That's probably the most basal holometabolous.
Isn't there? I don't know.
Honeybee, yes. MLHAV3 is the best annotated so far.
I think we're screwed. I think we have to go with Drosophila as the #1 choice.
Why? Go for B. You have 2 B researcher here.
Yeah, but there's just not been as much effort put into functional annotation.
I would say that
you do methylation.
I'm not sure.
I would say, I don't know, Herman can comment, but we've used Drosophila as a starting point and it worked pretty well.
Yeah.
Yeah, there's usually a lot of translative error if you're trying to jump between species. So and you're right, most of the stuff originates from Drosophila anyway.
Yes.
I mean, you'll get some from beetle that was direct and a few other species, but yeah.
Yeah. And Anna, I don't know. I know Ag100 Pest was working on generating new Apis mellifera assemblies. I just don't know and
Yeah,
presumably.
we have
this
better
is-
assemblies than what's available, but they're not out yet. We're doing all the breeding lines. Those assemblies are together right now, and then we'll be doing all the subspecies as well. And so it's a big giant pangenome project. But yeah, that stuff's not out yet.
Yes, I like that. Not biased, but. Yeah, okay, so Drosophila is probably where it's at. So then, yeah, you feel free to use NCBI or Flybase to the best of your abilities to find that reference data. Okay. And so again, I'm not going to, I'll talk to our team about that blast bug. I apologize about that, but again, if we can search for XP here in Apollo, I did have that identifier. We can go to that. And it's not called ebony, so it's mycosubtilin synthase subunit C. So this would be an example where it might be, if based on your further analyses, you actually do consider this to be the ortholog of ebony, we might want to do some nomenclature edits in Apollo. So. Okay, so that's the basics that I was going to go through of finding stuff. And then I wanted to start talking about naming, if I may. Sorry, I'm getting the spinning wheel. No, I'm going to turn my camera off to see if that is part of it. Well, my computer is not cooperating. Any questions?
Yeah, I have a question. So these are so far what you've shown is we're trying to see if a known gene exists in Schistocerca, and then we're confirming that.
Mm-hmm.
What if I have done some analysis and thing I'm interested is a unknown gene, and is there a way for me to figure that out directly in Apollo by comparing it with other known genomes in I5K workspace?
If you've got your, I assume, so what you're trying to say is you have something based on transcriptome data, so you have a sequence for it?
Yeah, that's a difference expressed, but it's unknown. How do I
Yeah,
figure
so
out
then
what
you
it
would
is?
take that sequence that you have for it and use the BLAST database to determine what gene model it would go to.
But we can do that in Apollo.
You would do it in BLAST, and then BLAST will have a link that'll open it up in Apollo for you.
Okay.
Well, no.
No.
So,
Oh, it doesn't anymore? Okay, I'm
Yeah.
maybe old on this. I thought that there used to be, but okay.
Yeah, so that's part of the problem with the new setup, and that's why I was directing you towards using that. So it's still BLAST, so you would go and do our BLAST and identify, go look through the BLAST results. So let's see if we can just do this here instead. So, and I'll go find that sequence again from odorant receptor, co-receptor, but I mean, the same concept would apply to any other sequence that you have available and that you're looking for in the genome. So here I'm using, I'm going to blast against the protein set, so I'm using BLAST P, and as my reference, I'm going to, so you can do it both ways. NR, if you do NR, that's going to allow you to search against everything, if you have a colleague who generated cDNAs from a different experiment that's external from your genome sequencing, that would encompass that. If you want to limit it just to RefSeq proteins, then you can just do search by RefSeq as well. And then limit it to, I think you can limit it to the genus possibly, but we're just going to go Schistocerca americana and limit it to that and do BLAST. Oh, so sorry, we can't do RefSeq select. Sorry, it's RefSeq protein. Okay, let's do that. I'm having trouble today. Good grief. Why did it not like that? Okay, there we go. So, but say hypothetically, instead of having this fly orco sequence, we had your transcriptome sequence or cDNA or what have you. So we just be blasting the same way here or at the I5K workspace. And so hopefully what we'll get at the end is a set of results where it may or may not be clear if there's you know, a single best hit or not.
So Monica, just to be clear, you're only saying that we can't do it at the I5K workspace because of the fact that it needs to be reset, but after that's reset, we could be doing this there, putting the transcript sequence in at the I5K database blast site, and then doing this search there, and then in the search results, don't you guys have a link that directly opens JBrowse?
So Anna, for our other organisms we do, but you might recall that for the Schistocerca, for the locusts,
We had to use the
sequences
bulk
are
blat
so
instead.
big that
Oh,
we are
okay.
not, yeah, we were not able to actually provide a blast against the genome sequence.
Okay,
And
okay.
typically our link outs that you remember from the blast results to Apollo, those are from genomes, and we were not able to provide that.
Okay, so those link outs wouldn't be there if you're just blasting against the protein sequences,
Yes,
which is what you can still do on the I5K blast.
exactly.
Okay, that's just where I wanted to clear, but it still would bring up the ID, and then you could put that in on Apollo.
Exactly, yeah. Thanks for pointing
Thanks.
that out. Yeah, so here's, I mean, here we have a more straightforward blast result set from NCBI. And so this is also perfectly adequate if our I5K blast is acting up like it is right now. And so you can just evaluate this as you know how to do as bioinformaticians. Looks pretty straightforward where we have low E value, high query, and you have this accession number here that I copied. And then you can, as Anna was explaining here, we can just, oops, I logged in, so it popped me to a different thing. And I'm logged in my like user demo account here where I already put something in. So here, make sure that it just starts with the same thing here. Oh, sorry, I should have done this. So the reason this is not working is that I'm looking at Schistocerca cerealis cubensa now, and this XP identifier is for Schistocerca americana. So log out. Okay, here we go. All right.
Yeah.
So, and so, Hojun, to your question as well, I mean, it's possible that you have a reference sequence and there is no protein that is a good, has a good blast result to this. In that case, you might want to search against the genome sequence to see if it's just not annotated. If the reference data that RefSeq used to generate the sort of homology-based portion of the gene prediction perhaps did not include that information that you have now, and therefore they didn't predict it. And so that case, you want to go to the genome sequence, and you can't use our blast to do that. You'd have to do that from NCBI. So that might be another take if you're going to like search your sequence space more thoroughly in that particular case.
So when we're looking at this page, can you explain what I'm seeing? I see the gene names, but there's a coverage thing and junctions. Maybe could you just briefly go over what these actually mean?
Oh, sure, yeah. So here we're looking at Schistocerca americana, the data. This is, here, I'll make it easier just to focus on certain things here. So here I have selected NCBI annotation release 100 and its genes. We separate out genes and pseudogenes just because they're Apollo won't let us load them in the same track, and they're just more easy to keep track of, but we can keep these both on if we need to. Looks like there's a pseudogene right here in the same gene space as the Orco. But yes, this is the RefSeq data that RefSeq generated mapped to the genome. So we took their GFF and loaded that as a track onto Apollo. And if you want to know more about where this came from, there's some information here about the data that we got. So I'm not going to go into detail in this webinar about sort of how to interact with this in Apollo, or how it displays exons and UTRs. I have that in the other webinars. And if we need to do a special session with this group to describe that, if it's not enough, we can try to set something up. I'm using these zoom in and zoom out keys to just sort of view the glyphs that represent the protein aligned, or this is actually, I guess, the mRNA aligned to the genome in a better way. But here you can see how the coding sequence is aligned to the genome. And if we browse all the way in, we can see this is where we're close enough that we can actually see the individual nucleotides of the reference sequence and their 6-frame translation forward and reverse complement. The tracks here, so GC content and gaps in assembly, those are ones that we generate at the i5K workspace by default. And this just shows you purple is GC, yellow is AT. You should have that information. Oops, sorry, got the wrong one. Away here about this track. So yeah, if you need to understand what the glyphs mean, we should have a track legend that explains those things. And gaps in assembly, you'll basically see a block that's black, where there's basically a base call of N. So those are those tracks. And then to go to the RNA-seq data. So this is the mapped RNA-seq. And when I usually look at these tracks, I start with the coverage plots. So there's not, Orco didn't have a huge amount of coverage here. There's more for the head, isolated head coverage here. But basically, these blue histograms show you the read density at a particular base pair in the genome assembly. So here, for example, I'm hovering my mouse over this area, and so here it says 15. That means that there are 15 RNA-seq reads aligned to the genome at this position. We have documentation on how we generate that, the RNA-seq plots here. That's a link to our GitHub. But it's important to remember that the data is normalized. So it's not the raw mapping. We actually do normalize it because we found that it helps for the annotation purpose itself. So it doesn't help to have wildly different displays of coverage if you're annotating. Of course, you would need that if you're actually measuring differential expression, but in this case, that's not usually what we're going for when we're doing Apollo.
But I guess, is it clear to anybody else here that me looking at this, I'd say that exon is well supported by the data from the isolated track, but using that crowded track, I would say that it's likely that there's an alternate splice form that entire exon isn't necessarily present in the transcript that's expressed in the crowded locus. Does everybody see that on that exon? But I guess if you're going to open up another exon, it looks like those all have full length coverage for both of those tracks. So it would just tell me that it's likely that there's a splice variant between the two, between the crowded and the isolated conditions there.
Yeah, that's certainly possible. What I look for there, and that's why I zoomed out while you were saying that, Anna, is to compare the coverage of the other exons, because if there's universally bad coverage, or not bad, but low coverage across the gene model, then I might be sort of wary of making that decision or conclusion, coming to that conclusion. But yeah, it looks like the other exons have pretty decent coverage, and so it's certainly possible that there's alternative splicing going on. The other thing to look at here is ISO head junctions. The junction reads, what these tell you, let me just get rid of the head to make it a bit easier to look at. So the ISO head coverage and ISO head junctions, those names mean those two tracks go together, okay? So this is showing the density of coverage of the RNA-seq at this location. And the junction tracks show the results of the, we use HiSat too, the HiSat alignment program in terms of where the splice junctions were computed. So here what we can see is that there's 7 reads, this value of 7 reads support this particular splicing event based on the HiSat mapping. That's what these junctions means. And so once you get used to it, once you get the hang of looking at these things, having these two tracks in combination really helps to see the validity of the gene models here, where you say, okay, there's support for this exon here, and there's support for that splice event based on these 7 base pair, these 7 7 splice events here going to this exon and so forth. So you see there's kind of a chain connecting all the different exons here. There's none right here. So this could mean that either the gene prediction is faulty or that there's just not enough RNA-seq data at this point. It's sort of ambiguous here. I would not necessarily jump to the conclusion with a lack of a splice junction read here that it's actually, there's a problem with the model, just because this level of coverage, 15 or 9, it's pretty low, you know. So if it was, you know, 100 or 1,000 here and then nothing, then I would say, okay, there might be a edit that needs to be made to the model.
But what you're saying here is that the numbers are low because they've been normalized and that sort of thing. So that low number isn't concerning to you. It's just been done so that you could not have the data, like too much data to where it makes it hard to browse it, correct?
Yes, that's true. But we do also, that's true. That is not a completely accurate reflection of the actual number of reads that originally.
But you still don't want wild swings. You don't want it to go from 10 to 0 to 10 or something like that, or 10 to 1 to 10 or something.
Yes. Yes. Does that make sense? Okay.
One quick question.
Go ahead.
On the ISO, it seems like you have 2 levels. Does this have any significance or?
There's 2 isoforms that have been predicted based on the NCBI model. You'll see that there's variant X1 and variant X2. So the NCBI has said that they believe that there are 2 different isoforms, and these are the ones that, based on their data, they have predicted. But you may find that you feel only one is supported, or only the other is supported, or that there are other isoforms that are clearly supported by the data. And so that's a decision to be made when you're annotating, is how many isoforms are you actually going to annotate at a particular gene locus based on the level of data you've got in that space, right? So we might say, based on that one particular exon, that if the start sites and things work out, that it would look like a viable protein, that we might add a third a splice variant in that place. Does that make sense?
Yeah, so what my question was more about the track that is called ISO head junctions.
Mm-hmm.
So when you look at that, it seems like there's 2 levels.
So those are just all those different ones. They just stack up when so that they don't overlap. But as you zoom in, they sort of spread out. See, so it's just so that they're not writing over top of each other, because one ends where the other one starts.
So there's no significance to that.
No, it's just trying to fit a lot of stuff on the screen.
I see.
But if you zoom in all the way, they might line up all together.
Yeah.
Sorry, so do those arrows mean that those particular alternative splicings go to the end of the gene? It's just not showing all the arrows because of the stacking? Is that what you were saying?
Which arrows are you referring to?
Yeah, so the ones that you're mousing over right now. Yeah, so these are different alternative splicing.
No, these are
right?
basically the introns. This is showing support for where the exon is cut, or not cut, but where the
I see.
exons are when you splice out the intron where the exons would be fit together, right? When we have transcriptome data, the introns aren't there. So when we
Right, so
align that
The
transcript to
exons
the
are those
genome,
red blocks at the top of that, right?
yeah, so the exons are those red blocks. They're different colors. I think you've got it turned on so that they're different colors depending on what frame they're in. But so that arrow in the junctions part is just showing you where the support is based on all of your transcriptome data for where the start and end of that splice junction is. That's what it means by junction. So where the splicing events would have happened. So I assume that they're stranded and that's why the arrows are going a certain direction. So it's saying that the gene is going it's on the strand so that it's telling you which side is the 5' and which side is the 3'. And I'm going to wind up getting dyslexic or something here if I start trying to say right and left in the middle of this, but without thinking clearly. But basically where that little box starts is where it's saying that, you know, that's where it feels like the beginning of that splice junction is, and where the tip of the arrow is the other end of where that support is.
Okay,
Right.
that all makes sense. But the very top, that's a representation of the genome, right? The NCBI annotation release.
So that's
100.
the representation of the gene model. So the red rectangle is an exon, and the lines are the introns that connect them.
Okay, and so are the two variants, the two different alternative
Yeah,
splicings
yeah,
for this? Okay.
Yeah, so there's the XM blah, 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 28.1 and 29.1, and it says at the end of the descriptive name, variant 1 and variant X1 and variant X2. So those are the two isoforms of that gene that has been annotated by NCBI.
Gotcha.
Or
And that was
predicted by
detected by the
NCBI.
transcriptome data, right? Not the,
So they use
Genome.
transcriptome data in their automated gene prediction process.
okay.
So there is evidence that goes into this, but there's also other factors that they use in order to determine that they use other protein data and other things, right? And they also used data that we don't have aligned here, so that might be a little bit jarring to you. But anyway, so based on what we know a gene structure looks like, there is an entire prediction program that decides that we believe that this is the structure of the gene, that there's 5 exons, that it's on the minus strand or plus strand, and that sort of thing. And so the boxes there are the exons, like I said, and the introns are represented by a line in between them.
Okay. Sounds good. Thank you for clearing that up.
Yeah, and so I think that this is where Monica was saying there's she has other webinars that teach you the real basics of moving around in the browser, what a gene model looks like, sort of how to read that gene model of 5' end, 3' end, what's an exon, what's an intron, where the coding portion versus the untranslated region is, and that sort of thing. So I think that that's not what today's talk is about so much, but is that right, Monica? That's what you would refer people sort of how to on how to get started.
Exactly. That's what those link outs are for. Yeah, I said I was not, I'm sorry, like based on the questions I'm getting here, I probably should have done that webinar with y'all first, and I can do that as well. If I mean, I wouldn't prepare any new material, but I just sort of walk you through those slides in addition, but it might be more worth your time just to go through either the webinar or the slides, familiarize yourself with your own data sets that are here with Shistasurca, and then we could have another Q&A session where you just go through and say, hey, I saw this in your webinar, and I was trying to apply this to this model that I'm interested in, but I ran into this problem. What does that mean? That sort of thing. That might be a more effective use of your time, but I'm open to doing any of those with you.
I have a question. So we're looking at this gene model. We've got 2 transcripts, and I decide I want to grab the protein sequence of it so I can make a tree with other species or the other things. So I'm assuming I would simply take the longest one.
Yeah. So if you're going to do a tree, Yeah, I'd suggest you use the longest one.
Or you use all of them and then you can,
Stick them all
I
in
mean-
the tree if I wish. Yeah. And then hopefully by this process, we would have found paralogs or orthologs, depending on how we feel. I am curious because looking at these, I wouldn't want to change anything, right? I didn't see any data that made me think, oh, I should change this. Although I guess I should compare the start of the protein sequence and the end of the protein sequence from Drosophila and make sure it looks about the right length, that kind of thing.
Yes. So, okay, so just briefly to backtrack, I was showing you all this page, which is the NCBI RefSeq annotation page for Cysticerca nitens, that data set, since there are questions about how that the gene predictions are generated, and this will tell you everything about what data was used in the sort of reference data based component of the annotation pipeline. It also has just a de novo annotation component as well, as most gene predictors do nowadays. So just so you know that that information is there at NCBI for you. Okay, so Fringy's question is basically also sort of a question about how you annotate, which is in the other webinars. But for this group, I would recommend doing multiple sequence alignment. So getting all of the proteins that you want for your particular question, and the fly ortholog, maybe one from Apis mellifera or Tribolium, based on what you were just describing earlier, and putting them into Clustol or whatever your favorite multiple sequence aligner is. And that will help you see, get a overview of, you know, how is my model complete? Does it have, it will give you a nice overview of, is there a methionine or whatnot? And is there a stop codon? Are there particular aspects missing? In one of the webinars that I have, I show how to go to Uniprot, which has a really nice overview of looking at the domains and understanding in fly usually, and understanding, you know, what's the domain structure, and then from that you can tell, like, oh, my model in Cysticerca is missing a bunch of exons with this domain. There might be a problem, or maybe this is some really cool biology going on, that kind of thing. So I encourage you to go back to those webinars and look at those. I will point out, so I am not even halfway through my slides and we're already almost at the end of the hour. So unless you all are able to meet for another hour, which I am, I'm happy to stick around, but if you're not, we need to schedule another session, because I have to go over this other stuff with you, I think, before you all start. And it might be helpful if we do have an intervening period where those who need and want to can go through those other slides and recordings, just to get like some backfill for the basic annotation process and what the tracks are. So maybe I'll break for right now. Well, I'll keep on sharing. I'll put my camera on and just ask how you all want to proceed. Do we need to stop at the top of the hour? Should we go on for another hour? What's your schedule like?
I could go on.
Yeah, I could go on as well.
Okay. Votes for stay. Okay, let's stay. And then if there's, I mean, we're recording, so I guess if there are people that need to leave, they can review this later as well. Okay.
Okay, I can stay, but I need 5 minutes to go to the bathroom and get some cookies. So if we're going to do another hour, should we make an official 5-minute break and then come back for naming and other things afterwards?
That sounds good to me. I mean, we could say at 3 p.m. It's 2:55 at least on my.
Yeah.
Sounds good.
Bio break time. Okay.
Yep.
Thanks, Shell.
All right.
Thank you.
I'm just curious, are there other, I mean, I know Anna has, but are there other folks on here that have annotated before using Apollo? Because I know that I have, and I assumed that there were other folks that had as well, but maybe not.
I'm guessing Fringy has.
Yes, yeah.
I've used another genome browser. I am sort of familiar with Apollo, but this will be my first go through, like, straight Apollo.
Sure.
Yeah. So have you done this, Amanda? Have you done this process with the GEP group yet, or have you not embarked on that process yet?
for Drosophila species, and they're like the distant, like Suzukiae and yeah, distant from Drosophila melanogaster. But I think I was going to ask about, it may be helpful for the Schistocerca group to bring in like the chain alignments from the other Schistocerca. I think that can be helpful once you're moving away from your model species. But yeah, the GEP, I was also going to evangelize a little bit for the annotation walkthrough that they provide, because it is very basic, but it's so helpful to have better familiarity with the process.
That's what I was just wondering is, I mean, we can have everybody, you know, plan to just watch the webinars, but I don't know that, I think there's something to be said for being able to just constantly interrupt with questions. And so I think that it might be also helpful to plan, like you're saying, Amanda, to teach them or to walk them through in a live session, the basics of what is gene prediction, what are gene predictions based on, you know, why are there splice junctions, you know, like what's a UTR, like all the really basic stuff that people know, but it's been a really long time since they were taught that, right? Because
or they don't know how it's represented in Apollo. Like, you know,
exactly,
you
you
want to
don't know
make sure that there's
sticks and-
evidence for an exon. Well, how do you make sure that there's evidence that this exon is being utilized? Okay, well, that's what these lines mean, because it's all just shapes and dots. And if you don't know what you're looking at.
Exactly. So I think that I think that's one thing that we might want to reserve a few minutes for today, is to determine how we want to teach this. And then I think also, Amanda, one of the strengths that I see you as having is because you've done specifically undergrad type projects where you had that learning goal, is that I know they had ideas at the beginning, but maybe you have some other ideas about ways to integrate that learning, or because I know, like you said, with your Drosophila, you were comparing these distant Drosophila, and then you were asking those students to make you had them doing some learning goals of that comparison. So I was wondering if you have some sort of insights about what worked and what didn't, what was painful, what you know, what was too much to ask, or maybe things that you think could be pushed further with more species or whatever. I think we've all kind of asked too about, like, what's the capacity of one student, you know, what's a good number of annotations to be like
one.
Yeah, well, that's exactly it. Yeah.
Have you also done it with undergrads, gene annotation?
Yeah, I've done it with undergrads. It's been a while, but I am refreshed to see that Apollo looks pretty similar to the last time I used it. So it hasn't changed too much. But yeah, I did stuff with the bedbug genome and the Hessian fly genome.
Cool.
Yeah. And Amanda will probably be doing at least 3 here, because you have to do it, as far as I know, your plans, you're trying to annotate 1 gene and at least 3 genomes, right?
Yeah.
Yeah.
Yep.
Yeah. Multiply everything by 3 or 6 or whatever.
Yeah, exactly. I would say, like, a good example is like 1 gene across 3 species, and then they can do the comparative thing and blah, blah, blah.
Yeah.
And then, Amanda, did you pair them up so that then they would have then talked at the end with whoever was the person doing that gene in the other 3 species? So then they put their trees together?
Yeah. Yep.
Okay.
Mm-hmm. Will you have a lot of undergrads in this, or is it going to be, I thought it was a mix, right? Yeah.
There's not a ton of undergrads, but I think the other challenge is that based on the backgrounds of some folks, right, and because they're new graduate students and things, I wouldn't necessarily put a huge differentiation there.
Okay.
And even perhaps, and I'm not trying to be unkind to anybody, but perhaps also with the skill and availability level of just about anybody in BPRI, there may be, you know, this is a whole new thing for a lot of people.
Okay.
Even some of the faculty have 0 genetics background, i.e. me.
It's the beauty of BPRI.
Indeed, you can get up to speed. All right, do we have everyone back? Looks like I see faces.
I don't know if we have Frenchie or if he's still in the cookie jar.
Oh, yeah. Well, hopefully he knows all this stuff. So, not everything though. There's some,
It's been a few years. I think the naming thing is actually going to be,
yes.
this was one of the biggest pain points all along. And I know you guys have worked really, really hard on this aspect. And I think this is as important about as getting the structure right.
Yeah. Well, while we're waiting for him to, and it's just riffing on some of the questions that came earlier about
I'm
the
here.
model.
Sorry.
Oh, he's here. Yay. Yeah, we just started. I just talked for 10 seconds. So in terms of like decision making about, well, do I need to modify this model? I mean, it sort of depends on what you want to get out of this exercise. I can imagine that for a, if your goal is to have a training exercise with undergraduates to learn how to make a phylogenetic tree, how to find orthologs or homologs across species, that kind of thing, you may not necessarily even need to make any modifications. You can go and say, okay, we're checking the gene structures here. We did a multiple sequence alignment. It's there. There's a start codon, a stop codon. Most of the sequence seems complete. And if there's a little bit of, inconsistency here, like there's, this track has two aligned reads, like what does that mean? You might not want to go into that level of correction, right? Because is it going to be worth your time to change the model and go into that level of deep detail? Maybe it is, but maybe it's not. If there's a glaring change, like I have some examples in the other webinars that you'll see where it's like really obvious, like that there's an exon missing right here, and we really need to add that exon or multiple isoforms, then it could be fun and straightforward. But if you're at a point where you're like crossing your eyes and thinking like, is this two base pair, that this two read alignment mean anything biologically? Like, no, you probably don't want to waste your time doing that. So that's sort of something that you have to keep in mind is the end goal of your annotation process versus how much pain you want to invest in using Apollo and doing the structural modification.
So my feeling for everyone is that in general, because the genome is such high quality, that the NCBI annotations are probably really good.
Yeah.
My worry is that some of the genes may be so long that we might be missing the ends of some simply because, it's a megabase long gene or something.
Yeah.
But I don't think that will happen, but that's my guess. Herman's been looking at a few genes. They are indeed long, right?
Huge.
But you did find the end of them okay in the NCBI annotation?
Yes. I've been using NCBI always for this. Not although in flies we use JBrowse a lot, which is the Apollo, the same as the Apollo platform. But and I do it with cDNA blasting, which works. But if you use Drosophila, you have to do the very low homology setting, and then it usually picks it up. And then I find the coordinates that way, and I'll type in the coordinates in NCBI, and then I have the gene, and then I download it so for me, I do all of this in SnapGene because I need sequence data for cloning, not for annotating. So I annotate because I want to clone and find guide RNAs rather than the other way around.
Yeah, so those perspectives of what are you actually going to do with the end result, those they help make decisions about how much of your time you want to invest in actually changing the model. Okay, so naming. And this sort of goes along the same question that was just phrased there, is like, what's your end goal? I'm gonna, as I said before, I want to devote some time to naming because I've seen a lot happen in terms of names, a lot of creative naming in Apollo. People get kind of excited about that. So I want to sort of first explain what the different types of metadata are that you can add to a gene that are used to describe genes. And you've probably heard of some of them, but at least when I started doing this job, I was incredibly confused about the different types of names and the different standards that were applied to them across taxa, because what is a name in fly is not necessarily a name in humans or in bacteria. So it's kind of the standards are all over the place. And so then also we'll go into questions of like, when is it worth changing the name or not? So here I'm going to talk about names and symbols and accession numbers, and I have them all next to each other just so you know that there are three different things here that you'll encounter. There's more than that too, but we're just going to talk about these three and what they are and where you might find them. So a name is probably what it sounds like. It's supposed to describe the function of a gene or protein. I've been talking about ORCO a lot, so odorant receptor co-receptor describes the function of that protein. Okay, I'm showing here on the right a screenshot of the Apollo 2 information editor. So Britt, you said you were familiar with Apollo from before. That part looks a bit different. That's changed since then, but hopefully you'll get the hang of it. And so here we're showing the kind of details about that model, and name is right here. That's where your name goes. Okay, I'll show you other places where you can find that too. Symbol is the short form of the name, for example, ORCO. So one thing that I show in the other webinars that I'll briefly allude to here is that you can annotate names at the gene or at the transcript level. So we can have multiple transcripts or isoforms per gene. And so the question is like, well, where do I put the name? You can have names for both the gene and the mRNA, but the symbol only applies to the gene. Okay, so here, if you look at the Apollo editor, you'll see the type is gene. You can show it later, and you can also see how to access the mRNA information, but you'll only be able to find or edit the symbol here if you choose to do so, which you may not want to do. Okay, and so accession numbers, and those are often called different things. So that's basically your permanent unique identifier for a feature. For example, XM, lots of numbers here. So some of the things that I was using to look up the genes in Apollo and JBrowse, those are handy because they're unique to that gene and that, actually in this case, to this mRNA in that organism, and you won't find that anywhere else. These are provided and maintained by NCBI, so we, even though it might look like in Apollo when you get in there that you can modify them, you can't. So we can't change, if you don't like that number, you're out of luck. They're maintained by NCBI. So just so you know, that's what those three things are. Next up, I'm showing you a screenshot, and we can go look at the actual browser there too, about where you can also find them looking in the JBrowse window that I showed you earlier. And this is where some confusion happens. So you'll find the name in blue and the accession number in black, okay? And here's another screenshot of how you might see this in an NCBI page. So here's the official full name of that, the fly gene in NCBI. So would you want to change this name? I don't think that in this case you'd need to or want to. If you decide, like Anna was saying, like, oh, maybe there's another isoform here that's undetected, you'd probably want to name that as well and probably call it something like odorant receptor co-receptor transcript variant X3 or whatever. So that case, you might want to give something a new name. Or in the other ebony example I gave earlier, so if you actually look and see and say, okay, we have evidence to say that this is likely the ortholog of ebony and we should apply that name to it, maybe the symbol, we can do that as well. So, but if you just think, well, I want to add a hyphen between co and receptor because I like that better, we probably shouldn't go to that level of effort for changing things. So, because it involves some pain to go back to NCBI to update those. Here just to show you again here, if we zoom in. Name, that's what we're calling the name and the accession number. And I'm going to some detail to explain this to you because when you add a new model to Apollo, it's confusing the default behavior of Apollo. When you add a new let me actually just do this, just so you see. So the default behavior is not the best, and it does confuse new annotators. While that's loading, I'll just go here and show that. So it will add, by default, when you create a new model, it will put the accession number in the name field for the gene, and it will create a new accession number with this 001. Okay, this is super confusing. Just to let you know, this is just the Apollo default behavior. This does not mean that we want this to be the new name of the model. And when I submit those, if we choose to actually move these to NCBI for archival and for propagation, I'll just remove those placeholder names. So just so you know, let's just show you, do this exercise. So I'm going to go here and I'm going to create a new gene model. So create new annotation gene. Super annoying. It calls it XM whatever. And if you go and look at this model here, this is all explained in the other webinar, but this is the Apollo the Apollo information, the editor. So you can actually change the metadata on this side and the structure up here. So, and it puts that in the name field. So don't be confused. Don't say, okay, we need to change this accession number. If you don't want to do anything with the name, just ignore that. And if you do want to change the name, you can just delete this and replace it with the actual, what we're calling functional name, odorant receptor, co-receptor, whatever. Okay. All right, so we're talking, okay, do you need to change the name? And if you've made the decision like, yeah, this name is wrong, or we need to add, for example, with the with this odorant receptor co-receptor, there's no symbol here. And when we had trouble searching for the gene in NCBI because this one wasn't called Orco, the metadata for fly it's all perfect and it's all there. It has all the aliases, and so you can search for all of them. We don't have that for Schistosarca americana. So you could choose to say, okay, well, I actually think it's worthwhile to tell NCBI that this is Orco. So that would be a legitimate use of using the symbol field and saying, Monica, tell RefSeq to put that in so we can index it.
So despite the fact that that would be a legitimate use, do we want to do that? And I ask because we could make a policy that if we only find a few things we want to change and update, we're not going to bother, right? And if someone wants to put that in the supplementary data of a figure, then that's fine. We could make another policy where we said for the educational purposes, we're not going to make any changes to NCBI at all. And that would make your life easier. And I think this group has to decide what kind of information is worth it, because I'm thinking in general, I'm more worried about students screwing things up or postdocs screwing things up, or more likely me screwing something up, than I am about missing some in what would be a percentage-wise an incredibly tiny update to their annotation.
Mm-hmm.
So that's my policy. I don't know how other people think unless something really amazing comes up. But certainly, you know, the kind of thing I can imagine doing though is someone finds there's 20 paralogs to a gene and they want to name them paralog 1 through 20.
Mm-hmm.
I'm not sure if we have a lot of that, then maybe, but maybe we have a policy where we just don't care and we just, you know.
Yeah. Let me add a little bit more context before y'all discuss that. But so as Fringy alluded to, submitting things to NCBI can be a pain. I will do it for you. And how that works, that process works is I have more slides about that because I actually want to try out a different protocol with y'all. But basically is when you all have decided these are the genes that we think are worth changing in NCBI, whether it's through their structure or their name or both, or just adding a new one because you thought there's a new isoform or this model was not in the reference data set at all, and we have evidence to suggest that it is. So once you've made that call, and I can help you decide if it's worth making that call, then I have to do one or two things. One is I don't QC the structural changes. There's really no good way for me to do that. I have to take your word for it that those changes are legit in terms of this is a new exon in the protein. But I do review your name changes, and this is why I'm going into a lot of detail with those as well, because that's where a lot of the pain happens. But I'm going to give you some guidelines to hopefully make it pretty straightforward for y'all. So once I've reviewed those, I can do two things. I submit the structure to GenBank. It's a process and it takes some time. If we need to expedite for publication purposes, I have some leverage with Terrence Murphy at RefSeq to ask him to ask the GenBank people to move it forward. So there's one thing. The second thing is, this is something I haven't tried yet either, but I've cleared this with Terrence Murphy as well, is if you just want to change the symbol or the name, I just put it into a tab delimited file and give the link to that tab delimited file to them, to RefSeq, and they pull it in. So it's a much easier process to change the naming, the symbol, gene, and product name, where product is the mRNA related name. So that process is simpler in theory, but I haven't done it yet because I just worked that out with Terrence. So there's different ways of doing that, and that's just sort of the time it will take on my end. But I'm going to do that work for you. So as long as we set this up well at the beginning, which I hope we can do, you can leave that aspect of the process to me. Yeah, I don't know if you all wanted to discuss that or just have that percolate in the backs of your heads while you think about what to do.
Ho-Jun's just got his photo up, but I was wondering if he can imagine a scenario where we'd like to change a name, but we'll have this discussion later.
Mm-hmm.
And then certainly, we don't want to change anything unless it's been reviewed by a faculty member.
Mm-hmm.
Not because we think they're smart, but because it's a double check.
Yeah.
What do you think, Ho-Jun?
I don't know. I haven't done enough of these, because it could have I mean, changing name as a taxonomist, changing name is always something that we need to think about seriously, because it affects the future stability of what we do. So I take, taxonomy and ontology and all those things pretty seriously. So it's not something that we can just change lightly because I feel like it. It's something that we need to think as a community, is it really valuable to do it? Or, for future people who are going to compare Schistocerca with other insects, is it going to make more confusion? So I think I don't really have a clear answer. I think it's case by cases, but in general, if there's already an established name convention for a particular gene, they will follow. If it's an obvious error, then we can fix it.
Mm-hmm.
Okay, so that's I think that's a good answer. So it's something we will try not to do unless we've seriously thought about it.
Mm-hmm.
What about uncharacterized loci? I've encountered a lot of strong marker genes that just don't have any homology to any known species, even after I tried blasting them.
Yeah, you'll find a lot of those. Attributing name to it really depends on the function, I think, and unless we actually know the function of that particular gene, all the names that we have is somebody's dissertation project. They figured it out, the function, and we're not there yet. So I'll be actually okay with leaving it as LLC something, until we have an additional proof that is involved in something, like some phase related protein number 1 or something like that, then we can name it later. But I don't think, if there's no obvious relationship to known genes in the GenBank, then we shouldn't really name it. That's just my sense.
Mhm. Yeah,
Where are you getting these sequences from?
Me?
Yeah, David.
Yeah, so from my presentation the other day, some of those clusters I showed you, some of like the top expressed genes in them are just, unannotated,
And
unknown
these are
loci.
unannotated in Drosophila? Or
They're
they,
just in the automated NCBI annotation, all these gene descriptions like that odorant receptor, coreceptor. I use the GFF files as my annotation files, and some of them just don't have any information. And even after blasting that particular unknown sequence, I still don't find anything.
for
So,
example, like this uncharacterized loc.
yeah,
or-
exactly. Stuff
Yeah.
like that.
Yeah.
so I think even in Drosophila, I don't know what it's down to now, but at least a third of things are uncharacterized functionally. Maybe Herman has more up-to-date numbers. I wish I could say something more useful, like perhaps if we blast the genes either side, you might be able to at least find orthology to other species, but I think everything's too far away for that to work. And then you've got, even if you do, you've got genes of unknown name and unknown function in multiple species, which isn't more useful. It's just a bigger pile of questions.
How do people go about, answering what the function of that gene is? I mean, the only thing I can think about from a bioinformatics standpoint is to throw it into AlphaFold 2 or something to try to get a preliminary structure and go from
In
there.
Drosophila and other species, they knock out every protein in the genome, and then they look at it and they say, can we see a different phenotype? And then on top of that, of course, you know which tissue it's expressed in. So if it's a neural gene, maybe there's a nice story from Daphnia where there's a lot of genes, nobody knew what they did, but if you threw the Daphnia in some funny polluted water, they'd suddenly get turned on. And so people didn't know what they did because they were never in that environment. But experimentally has been the answer, and that's why we transfer everything from Drosophila, because they did the most experiments.
Gotcha. So you would just have to do a forward genetics screening, basically.
Yeah.
Okay.
But that's
But
also the problem with, Drosophila, because obviously the functions can be quite different, and you have to, that's one of the things you always have to be, that's what I talk about translative error. You have to be careful. It's one of the problems with KEGG and other things when it assigns functions, right? It'll tell you it's important in gill formation. Well, that's nice. My species doesn't have gills, right? And this and that. So you really have to be careful with how, what the process was and what evidence was available, how much similarity, was done to transfer that knowledge to your species, or that, that annotation to your species.
And as Ho-Jun said earlier, we've got a bunch of unknown genes with differential expression based on phase. And so you could call them swarming phase genes, right? In a paper later on, but that's probably not quite enough.
Yeah.
I'll show you some rules later on that shows that no, you can't, like.
Okay, so we'll let Monica get back to this.
Unfortunately. But one thing, David, and all of you too, so I wanted to point out for this, all of the, so we ran our functional annotation pipeline on all of the protein data sets here. And so I showed you the links to the flat files where you can essentially just see like a GAF or Excel file that shows you, here's my XP number and here's the Go or KEGG number that was associated with that, and you can perform analyses on this. But this is also in the browser. So what I did here, going back to our Orco example, is if you control click on a Mac or right click and go to view details, this shows just a lot of information about the data in Apollo, and we have an ontology term category that shows you the gene ontology terms this was annotated with. So if your protein had similarity based on the annotation pipeline that is referenced in the page that I showed you earlier, then this will show up on the browser page for that as well that way. I mean, I just searched for some uncharacterized ones here, and I didn't find, there was no gene ontology annotations for that. And this is an XR too, so that it's not a protein, and so by definition, it won't have. Yeah, these are all XRs. So yeah, so not all of the proteins will have a gene ontology annotation, and sometimes it will be uninformative too if it's protein binding, like, yay. Sometimes there's information there that you can use, and there's also KEGG-based information, which, as Anna says, you got to take it with sometimes a shaker full of salt. So, okay. So, okay, so if you actually do choose to change a name, and in the case of uncharacterized whatever, unless you have good experimental evidence where you think, yeah, I think I can change this, you probably don't want to do it. But if you do think you have enough information, then here are some guidelines. So if there is a homolog, then it's a good idea to adopt that name. You can reuse existing established names. I had a conversation with Terrence about this. So you mentioned earlier calling it like swarming phase gene or whatever. And fly, they love adding, funky, interesting, cool names that are based on phenotypes, and that's totally legitimate in fly, but nobody else in the genomics community allows you to do that. Humans, for sure, not like on the human genome and whatnot. So the general guidelines for that we're applying is you can't use a phenotype to describe your gene. But if fly already did it, then go ahead and use that name, and we can put it in there. So there was a question earlier about like, well, I don't want to change the name, but if there's a precedent for having that name in a different species, and you think it would be useful to maintain that, we could also entertain the thought of adding that as an alias, and that wouldn't be the full name, but we can put it in as an alias in RefSeq. I think they'll let us do that. So, Ken and Barbie, oh gosh, yeah, that's a good example. Yeah, I'm not dead yet. So yeah, you can imagine, I think Anna pointed this out to me once, that if you had those sorts of gene names in like the human genome, that some people who actually have deficiencies in those genes might not be super happy about the name of that gene. So
yeah, there's been a whole thing about changing those fly names for quite a while, because they don't really want those propagated to human disease.
Yeah.
Humans are boring.
Yeah. They're boring, but yeah, that's unfortunately the way it is. So yeah, so you can reuse existing established names like odorant receptor, co-receptor. Some groups like adding a species prefix, like especially in this group, I imagine if you're writing your paper, you want to distinguish the Schistocerca americana from the Schistocerca piscifrons one. And how do you clarify that in your manuscript? So in your manuscript, go ahead and use species prefixes, but we can't submit those to GenBank. So if you add them in, then I will remove them, basically. But in your manuscript, do what you need to do. If you're not sure, you'll see that NCBI or RefSeq adds a lot of -like to their names. And if you want to imply uncertainty, you can do that as well to your name as well. So you can, here's a ultraspherical example. So ultraspherical, good. Ultraspherical like is okay, but don't call it CLEC, ultraspherical, or similar to. They don't like that. And when I review your names, I can go through all those things too.
What do you recommend for a paralog if we find there's 3 genes in locus and only 2 elsewhere?
That's hard. If it's a gene family with an existing naming convention,
yeah, that's the only ones we're gonna find.
Follow those naming conventions, and I can point you to some of the documents that are available for that. Or we can even, I don't know if this group is doing cytochrome P450s, but we could talk to David Nelson or others about that. There is some odorant, I think some of you are going to work on odorant receptors or odorant binding proteins. And so David Mitchell, since Hugh Robertson retired, David Mitchell is my new go-to person to ask about that. And so we could talk to him and loop him into the conversation about naming for those things. In terms of if there's not a defined schema or defined way to do that for a gene family, you can make one up, I guess. You might want to do that append-like thing, but if it's necessary for you, and if for your research purposes, you do need to start keeping track of the numbers, then do I have this in here? Yeah, here, if you're naming a gene from a gene family, here's the list of naming systems. And otherwise use Arabic numbers to specify the different numbers. Often people will just go along a scaffold and say 1, 2, 3 based on position left to right. And with your genomes, since they're already at chromosome level, a lot of them, you might actually be able to do that and have that numbering preserved for longer, as opposed to the previous assemblies where it was, thousands of scaffolds. And so it was just anyone's guess as to what those numbers would be once you actually get a correct assembly. Does that help answer your question?
Yeah, thanks.
Okay. Okay. Yeah, if you're naming an isoform, I say here use isoform A and isoform B, but actually I'm going to retract that. When I send you these slides, we can just do transcript variant 1, 2, 3 like RefSeq is doing, because we're submitting these names directly to RefSeq. So I will correct these things later on. Hopefully you won't be dealing with fragmented genes. But that was a previous assembly with previous generation assembly problem. But if you do have a fragmented gene and you really spent a lot of time figuring out here's the first part and the second part and the part in the middle, put that in the comments and we'll try to propagate those as well. And don't add partial or part of to the name. And then, yeah, so here's the case of well, I have a new gene and I know that it's associated with swarming. But that's a phenotypic based description, and that's a no-no, unfortunately, for if you're not working on flies, on Drosophila melanogaster in particular. So if you can say the litmus test is, could you propagate this to a new species that doesn't swarm? And if you can't, then don't call it that. So for example, magnesium transporter, that's a decent name. Diapause associated protein. Yeah, sorry, we can't do that. Okay, we talked about naming genes from gene families, so covered that. Okay, moving on to symbols. So I talked about that briefly earlier. So it's a short form of the name, for example, Orco. It's assigned to the gene. If you feel like you need to get fancy and assign a new symbol, let's not do that. I'll just say, no, we can't do that. So it's okay to adopt it from a homolog, which is going to be fly, because they're really the only species in arthropods, let me not lie, in insects at least, that don't have a naming authority. But so this is just going to be transferred via homology or orthology. So you can add a symbol at the gene level if there's an existing one in an ortholog, for example, the Orco one that we mentioned before, and this might help with searching this in NCBI in the long run. Yes, so that's basically what I just said. The prefix rule applies to this too. If you want to use prefixes in your paper, go for it, but please don't apply them to Apollo. All right, so that was what I was going to tell you about naming. Before I go to just sort of the end of your process and how we're going to deal with things when they come out, are there any questions about naming yet so far? Let me check the chat. Yeah, fly names. Okay.
So in terms of naming, and you laid out do's and don'ts. Is there an actual guideline or rules? I mean, for naming species, we have a ICZN actual guideline. Is there something like that, so that if there are some cases where not quite sure, can follow?
So here's our page about naming guidelines. So there's no official naming guideline for insect species other than what I've produced, which is not official. I have in the back of my head that I need to write a grant or get somebody else to write a grant, and so we can actually have a nomenclature committee for insects. But so what we've used instead is following the international protein nomenclature guidelines. So those exist for proteins and on a general basis, and I think the link out here should hopefully work. Yes, to this PDF. So this is the document that we use for protein guidelines. proteins can't have symbols unless you're a prokaryote, but for eukaryotes, they can't have symbols. So for those, that level of information, we leaned on the human genome nomenclature guidelines for those. So this should be linked to in the slide. So if you have questions, you can refer to this document. And you can also just ask me as well, and I can help you interpret that.
It would be a good time to have that discussion globally because the EPP and EBI is going to be punching out a bunch of welcome annotations. So maybe write that grant this year.
Yeah, or just do what they want to do and hope that they correspond with RefSeq. That they're the main data generators and annotation generators nowadays, and so they're basically going to be the authority for generation of those gene names, and we'd have to work with them. But the other question is, so once we get to the point where we're like, we have the tools for that Fly used to with these excellent genomes and with CRISPR-Cas9, where we could actually go and determine function. That's when we're going to start having these really important questions for those uncharacterized ones. And so that's still there. Yeah. Okay, so all right, annotation outcomes. So what I was thinking about for this group is leading back to the original goals and outcomes that we expect from this group. So this is actually, and this is something that I thought new about for you all, because this is the first time I've actually had an annotation jamboree where I think we have enough people with a defined enough goal that we can actually get these things out quickly and possibly have a decent turnaround if you decide to submit to NCBI to get you accession numbers, because that part takes some time. So I actually am going to experiment on this community, if you will indulge me, and hopefully it won't be too hard, to see if we can get a model like this to work with you all. Okay, so here's what I want to ask you all and to ask you to think about. So say you have your odorant receptor co-receptor and you decided to add an exon and you decided to add a symbol to it, okay? So the question you ask now is, well, do I care enough about this change, these two different types of changes, to talk about them in a published paper? Or if I don't want to do that, is it worth for when I have students or other colleagues working on locus to propagate that to NCBI so that when they download the next annotation release, they don't have to deal with the pain I had to deal with to realize that that exon was missing, okay? So if to those questions you answer yourself yes, then yeah, then it's worth engaging in the process of moving those gene models back to NCBI. If you say no, if you think it's not worth it, so there's one or two options. One, we can just delete stuff from Apollo when you're done, and it will vanish from the face of the earth. And that might be a reasonable outcome if you're not publishing it and you really don't need to propagate that work. Or a hybrid option is we can submit just your FASTA files and whatever else GFF files to the Add Data Commons, so they live somewhere. This is like a repository like Dryad, or you can use Dryad or whatever. And then that will live as a FASTA file there. Probably nobody else will find it, but at least for data management purposes, you can say, well, it exists on the internet somewhere. So, but it's not going to be really retrievable or reusable that well unless it goes into NCBI. So, with those questions in mind, I created a new status field. So this is a field that you can select where you can choose, I guess, 4 different options here. One is if you change the name, you click submit name and submit name to NCBI. So that means you're like, yes, I want this to live on, okay? If you just submit it, change the structure. If you just added an exon or added a model, submit structure. Or if you did both, do both, okay? And if you do that, that will signify to me at the end of this annotation effort that I need to get moving on it and submit it to NCBI. This delete option is just an option if you actually think the model is wrong and needs to be deleted from the gene set, but hopefully you won't encounter that too. I can show this to you here as well. Status here for this gene, and I can say, I changed the name but not the structure, so I'm going to add this status to this area, okay? And that will make it a lot faster for me to go through your data, your annotations later and say, okay, I'm just going to look at these things and not the ones that don't have a status. and then we'll submit those to NCBI. But that also requires a bit of care on your part to not forget to do that. So, and if you say, well, we can't, it's hard for us to remember, then I'll have to think of a different way, and it might take me a lot longer to interact with you. I created a decision tree for you all, so we don't have to go through this now, but if you're asking yourself the question, what status should I use? Here's a decision tree for you to see. So are you going to refer to it in a publication? No. Well, does somebody reuse it? No. Then leave the status empty, and so on and so forth. So hopefully it's straightforward enough where you won't need this, but this is there for reference in case you do actually want to use that field and want to reuse that model and submit that further on. Okay. I guess maybe to refer back to some other things here. So will you refer to it in a publication? Yes. Did you change the structure? No. You change the name? Yes. Okay. Submit status to name to NCBI status, that kind of thing. Okay. The next question that I thought that you'll need to ask yourselves is, so I'm writing a paper about this. Y'all are probably going to write papers about the genomes with and refer to specific models in the papers, right? Okay, not just your phylogenies. And even for those, maybe you'll want to use these guidelines. So if you're referring to your model in a publication, in the past we've had trouble because if you change it, then what's the accession number? If you change the structure of the model, that will actually, once we submit that to NCBI, that's going to trigger a new build and it will have a new accession number. Okay, and so the question is, well, what do I do in the paper, right? It's a changed model. So in this case, I think we can have the turnaround fast enough that by publication time, I hopefully can get you a new accession number and you can refer to that in your paper. If you just change the name, the accession number won't change, so that's not a problem. So that won't be a big deal, and you can just refer to the accession number. So I have another decision workflow and some guidelines for you all that you might want to adopt, that I suggest you adopt for referring to changed gene models in your publications that you publish later. So if there's naming changes to the model, yes. Structural changes, yes. So I recommend that when you draft your paper, like before it goes to like being published and whatever, refer to it by the new name you gave it that's approved by me, okay? So it has to be approved by me first. Then, hang on. Structural changes to the model. Oh, I need to update this, sorry. I will review this too, but we can also provide that accession number to that model as well, because that will be a new submission to NCBI that will get a new accession number. But it might just be the easiest to call the new name because that will have to be available as well after submission. So and so on and so forth. So in the case where if you're having naming changes to the model, but no structural changes, you can use NCBI's accession number and optionally the new name. If there's no naming changes, but structural changes, use the original accession number. This is the clicker here. So if you use the original accession number and call it modified, okay? And I know you'll forget this, and this is why I did this decision tree. So I will provide you with these slides and also help you think through this later on. And if there's no naming changes to the model, no structural changes to the model, use the NCBI's accession number and optionally the name. Especially, and if the name is not unique, definitely use the accession number.
So I just want to point out to everyone that what Monica's suggestion here is actually pure magic, and it was been impossible for many years. And in the past, the only hack I've seen that works is people literally printed the sequences and names and the supplementary data of anything new they have, so at least whoever read the paper could get it. But thank you very much, Monica, for even suggesting this, because this is well beyond the call of duty.
Well, I feel bad about all the I5K pilot projects where there's a lot of data that just went to die in the supplementary materials. So, and some of it was hopefully it's on water under the bridge now because better assemblies from the I5K pilot. But that's what happened, because it was just impossible that we hadn't made the link to NCBI yet, and it was just impossible to get the G models there. And
yeah, let's not worry about the old ones. We're in a better world now.
Yeah. So hopefully, especially if this is a limited group targeted, We have connections to NCBI now, and if GenBank is having a rough day, I can hopefully pull some leverage with RefSeq and say, hey, can you ask GenBank to do this by your publication deadline? Hopefully we can make it work, and we can see if this is a good model for you all. Okay, so that said, so annotator emails, I will make sure to save this chat before I leave the Zoom, because that's where a lot of the emails came from. So hopefully I can do that. I mentioned we're missing the Gregaria, RNA-seq, and protein alignment, so we think we covered that. Keep in touch with me if you have any questions. If you want to set up another Q&A session with me before your you go ahead with your undergraduates, or if you want to have me sit in on any sessions with them, let me know. And hopefully I can manage it time-wise. And then based on that procedural stuff that I just said in terms of publication timing and NCBI submission, it is sort of time sensitive. And so if you could keep me in the loop and say like, okay, we're done with round 1, we know what we want to submit, we know what we want to publish, then let me know. And we can make that iterative if we need to. Okay, so we can just talk to see what you all need. So this is, yeah, both for the annotations and then publication timelines for any manuscripts that come up. Okay, that's all the scheduled programming that I have. So what other questions do you have right now? Other than can we be done, because we went over about an hour over.
You said you'll be sending these slides out, or was that the link you already posted earlier? Is that a different one?
I haven't posted them, so I'll
Okay.
send them out to you afterwards, and I'll post a link on the I5K website too, I5K Workspace website.
Okay, sweet.
And I'll talk about getting that blast problem fixed. So,
I have a question about using BLAST in Apollo or in the I5K workspace. What are the benefits from your perspective?
yeah, I mean, it's if you really just want to go use our sources and just be focused and limited to the RefSeq proteins, I think it's a little bit cleaner. But honestly, since we don't have that link out anymore, potato, it's just whatever interface you like best. You know, the one other magic trick that I should have showed you that I didn't is, especially in Hojun's case where, you know, you have cDNA or protein and you can't find any decent hits in the protein set, and our genome link outs don't work so well. So you can use BLAT in Apollo. And the way you do that is by using this search tab. There is this button that says all genome sequences. I beg you not to click that button because that it just uses a lot of resources, and the machines that we have, since we don't have, you know, we don't do heavy computational work on the servers that these things are on, they're not really equipped for that sort of search. But if you have, you know, if you know that your protein probably aligns to like this genome area, then you can just you be in the sequence that you want to be in. So NC, if it's NC blah blah 119, stay in there. Put your nucleotide sequence in here or your protein sequence. It doesn't like the identifier. So let me actually get that sequence here. Where was that? Let's see if I can break our servers. So here's our fast file. So what it doesn't like is it doesn't like the identifier line. So you have to take that out. Otherwise it'll tell you it fails. That's just a way Apollo works. But you can hit search. Oh, and what I did wrong was I didn't select BLAT proteins. So I got to do that here. Great. I'm having a lot of live demo problems today. Apologies. Let's see. Yeah, I don't know what's going on here. So I got to talk to my team a bit more about what's happening. Anyway, so that should work. I gotta talk to my team about why not. But in theory, that will allow you to search for the genome for that particular query sequence, and it will show you an alignment, a highlighted alignment on JBrowse or on this particular part of the JBrowse Apollo viewer. It won't show you, it won't create like a little glyph for you that you can use as annotation evidence, but it will show you where that alignment is by highlighting.
So Monica, I think that's actually a good point. Let's say somebody feels like something is glitching or broken or whatever. Who should they write? How should they contact that person, and what information, screenshots, etc. should they provide, sequences, species names, whatever? What should they provide so that you all can hopefully best either recreate it or realize that just blast is down or something like that?
Yeah, that's a great point, Anna. So, I mean, you can email me, but I am not always there. Like, I'm going to be off on vacation next week. So if you email me next week, I won't answer. So in that case, I'd suggest you go to our site and use our contact form. And just add your information here. That basically goes to Anna's husband, Chris, and myself. So you'll also get Chris online, and he'll be able to address questions as well too. Yeah, so but to recreate it, it does help to have, I mean, I guess you can't attach a screenshot here, so that would have to be in a message to me or to Chris for that. But if you just give us the URL where you see the problem and just describe, you know, I tried to log in and it was this link and it didn't work, or, you know, I did this query in BLAST against Schistocerca gregaria and it gave me no hits found or whatever, that should be sufficient, and we can always follow up with you. That's a good point.
So I have a question. So we didn't talk about building a phylogeny. Is there a way to directly download a particular sequence from Apollo?
Mm-hmm. Yeah, so I would do that by, let's go back to odorant receptor. So you can do that by dragging the model up to Apollo, like I did here before, and control click and get sequence. That allows you to get the sequence in different ways. It can be this peptide, you can get cDNA, CDS, and if you need like a particular snippet, plus or minus a couple of bases, you can do that.
So it just automatically takes out the intron and just does the exon.
Yeah, for these different types, yeah. So peptide sequence, it would just give you the translated protein from the CDS without the introns. But genomic sequence, that would give you everything like from the start to the end with the introns in it.
I see.
Plus flanking sequence if you want for this particular option. And you know, I can ask Chris too, because he came up with a neat way to use Apollo's web services to get a particular region from of FASTA sequence from an organism. Then you just paste that you format the URL. It's URL based, so you format the URL and paste it in your browser, and that retrieves a sequence. So I mean,
it'll be useful. Say, you know, I want to build a phylogeny of Orgo. If I can download Orgo sequences from every single species that you have on your database in one shot, and then it just sends me on a line FASTA file of everything. That would be very useful.
Yeah.
We're going to have to decide which sequences we want, so everybody just doesn't do different ones, and then we have to repeat it all.
Right.
Yeah. You know, I recommend that for that you use NCBI. So we thought about having resources for that kind of thing for a long time, and we decided not to because NCBI already does that, and there's already we use the RefSeq proteins, and so I would recommend, you know, you can pass a list of identifiers. So say, you know, you've identified, I want all these different XP accession numbers and get the FASTA sequences from that. I've used batch entree for that actually. So
Mm.
if I have that URL up, where you just have, you make a text file of all those different identifiers and you patch it. If you've already done this, apologies, but if you haven't, just briefly, you have to select the database. And so if you have XP identifiers, those are protein identifiers. So you choose the protein database. If they're XM identifiers, those are nucleotides. So you choose the nucleotide database. If you do the different one, it will fail. So XP, and then you just select, and it's just, each line, I think it's new line delimited, has your XP identifier, and you hit retrieve, and that gives you a list, and you can select all of them and say export to FASTA. Boom. So that's how I'd recommend that you do that. And we made the decision just, it would be cool for us to have that on our website, but developing software on the USDA is hard enough. And so we figured they're already doing it. Just point them to.
Yeah, that makes sense. Makes sense.
Yeah.
So I have just one technical question. Not many people might be interested in this. The pseudogene layer, what are those pseudogenes?
Let's see. So those are gene predictions. Let me clean this up a little bit so they're easier to see. Those are gene predictions that NCBI provided, where their gene prediction program decided that these are pseudogenes. And so we don't have the link outs to that. The source, if you look at the information about them, it's their CM search program. So I'd actually have to look in to see what that particular aspect is too of the pipeline.
Okay.
They give you a breakdown of the
Okay.
type of pseudogene in that sense. I mean, with pseudogenes, it's without having additional functional information or annotators go in, it's kind of hard to know what type it is, right? If it's, just the genome sequence is poorly predicted, and therefore it's actually just a broken gene model, and it's truncated at the beginning of the sequence.
I see.
So is it a pseudogene because it is transcribed and it's partially transcribed, that kind of thing?
Yeah,
so.
I see. So, I mean, I asked because I'm also interested in nuclear mitochondrial pseudogenes. So I know that Schistosarca genomes is full of nuclear mitochondrial pseudogenes. And I was wondering if there's an easy way of searching for it in Apollo. But
Well, let's see. So
we'll probably search for like CO1 gene
Yeah.
against the nuclear genome.
So there is, you all do have a mitochondrial genome in this assembly, right? Do you remember what the identifier is? I guess we can go and look. Actually, let's just go check.
Before we go too far down this minor research project, do we need to meet again? Perhaps not with Monica, but to settle on dates for when we're going to do this and do a bit more organizing, and so we can get everybody can show up at the same time. And then we also need to decide what else is remaining to do. Maybe we need to get someone to generate the protein sequences. I guess that's just a T-blast-N against maybe 5 or 6 species of proteins, like certainly Drosophila in human and mouse and yeast and tribolium and honeybee and hopefully a termite and a cockroach. But we need a date for when we're going to do this. I think we pinned it down to a week, but we can sort that out later, but we do need a meeting for it.
Well, John, how about next week sometime?
Yeah, I mean, next week will work.
You guys just work it out and then tell us and we'll show up.
Yeah, we'll do.
So here's the mitochondrial sequence. I got to that by
Yeah,
the information page has that here, and that gave me the identifier. So I don't see that any of these have particularly useful names given by RefSeq. You could try to go to the NCBI name search, but again, the best way for you to find out if they actually gave these proteins names, other than looking here and seeing that they didn't,
yeah.
is to use a blast-based approach to at NCBI or with us
Mhm.
and see what's there.
Yeah,
in the
okay.
search results.
Okay, that could be a side project. Right, sounds good.
All right. Anything else? No?
So this has been hugely helpful. Thank you, Monica.
Thank you.
Yeah.
Yeah, thank you,
Yes.
Monica.
Thanks.
Sorry about some of the technical glitches, but I have a couple things I know I can follow up on and see if we can improve them before you actually start for real. So,
Thank you.
okay. All right. Well, keep in touch. Let me know if you need anything. Actually, how do you download the chat or, okay, save chat. Got it.
I actually made you the list also.
Oh, great. Thank you. Appreciate it. Okay. Well, take care.
Sounds good.
Have a great
Right.
afternoon.
Thank you so much.
Bye, everyone. Thanks.